Hi, all. Welcome to What's New in MPMI, the virtual seminar series of the MPMI Journal. I'm Jean Harris. I'm the host and organizer of the series. And while people are entering our virtual room, why don't you type into the chat where you're coming from? It's always fun for us to see where everyone is coming from. So I'd love to see that. I can see a number of people already joining our room. Claudia Martinez from Mexico, Puria and Safi from the US, Valdir Moreira from Brazil, welcome. Moving fast. Xian Yong Wei from the US, Noma Chingandu from Ontario, Zhao Li from Fargo, Florence Wisniewski-Dai from Lyon, France, same lab as Asna. And we have a number of people from France, Claire from France, Ye Jia from Ohio, Mathieu from France, Anil from University of North Texas, and lots of people coming on. So welcome, great to have you here. We're using the, we're using that chat just for the welcome. We'll move on to Q&A later. Anyway, welcome. This is What's New in MPMI. I'm Jean Harris. I am the host and organizer. And I'm excited today to present a really interesting talk about a paper that caught my eye right as it was coming through. But first, let me introduce who's here. We have Siva Sankari is my co-host. She's from the Stowers Institute. And Siva, do you want to introduce yourself?
Hi, hello everyone. I'm so thankful to Jean for making me the co-host. This is a paper that's close to my heart now. And I just opened my lab very recently at the Stowers Institute. And I'm also closely associated with the journal MPMI because I'm an assistant features editor in the journal. So as a group, we do science communication and take the papers and bring the human part of it. And we also write commentaries and something to show how the paper is actually done and to make it accessible to everybody, because the manuscript is like fully packed with science. We want to make the manuscript accessible. And so I want to pitch in for something we do today. So we also have a podcast coming along. This has always been there. It's called Microgreens. And we have podcast on diversity, on science, on faculty careers, on many different things. So I would highly encourage you all to, when you find time, when you're driving, when you have some time, listen to our podcast so that you will get the other side of MPMI as well. And with that, I will pass on to Jean again. Thank you.
Thank you, Siva. And I'm also glad you talked about it because we're looking for new assistant features editors. So if you are interested, I'm not sure where who you email it to if you're interested, but you can always contact Siva and she can tell you.
Yeah, I can definitely get you up. And there is also in the MPMI website, there is the application that you can fill in. And the last date is on Valentine's Day, February 14th. So fill in, if you are interested, fill in the applications by that time. And you will love it. I thoroughly enjoyed being an AFE, and getting this network and connections is awesome.
Thank you. Yeah, so I should say that this is a position for postdocs and is a volunteer position. One of the things that, and it links actually very well to what we're doing here with our What's New in MPMI. One of the big focus, when I started this series, a big focus was inclusion. We have a very international society. this allows people around the world to connect in real time with the authors. And this assistant features editors is another way for us to produce content where we can connect around the world. And this has been a real pleasure of this, to see so many people returning and always new people coming to watch these seminars. So today I have Hasna Boubakri from the University of Lyon and Melanie Gasser, the first author of a very interesting paper on a complicated interaction. So this is an interaction between the roots of a woody plant, a long-lived woody plant, a tree, and an almost unculturable microbe, Frankia. So this is an incredibly challenging system to work in, and yet important nitrogen-fixing interaction. So how do you even figure out what's happening inside the cells, inside a root of a woody plant? And so it's a fascinating story. And one reason I loved this right from the beginning was that they've actually identified a little bit, so we know a little bit more about what's happening at the point of contact between the microbe and the plant host, not only outside, but also inside the cell. So super exciting new work. And I think that Hasna is going to tell us a little bit more also about what direction this research went. So very exciting. And I am going to now turn it over to you, Hasna, to tell us a little bit about yourself. And why don't you share your screen and we can transition to the seminar.
Okay. I will share my screen first. So good morning or good afternoon, everybody. I will share my screen. No, it's not this one.
Yep. Nope, that was okay.
No, not this one. I think so.
Oh,
this one. This one. Share. Is it
yes.
Okay for you?
Perfect. Okay. Thanks, Hasna. I will turn it over to you.
Okay, thank you very much. Thank you, Jane Harris, for inviting me to present my work. So first of all, I would like to introduce myself and to tell you a little bit about me. So I'm Hasna Boubakri, assistant professor at the University of Lyon. I'm sharing my time between teaching and research in the ecology microbial unity in Lyon. This unit is made up of 7 teams working in microbial ecology in different ecosystems, such as urban, agricultural, and natural ecosystem. We also have a great experience in study the insect or plant interaction with microbe. And we have also the capacity to identify the microbiome and their function in different matrices, such as soil and host, and to evaluate the ecosystem service brought by this microbiome. So I have a background in molecular biology, metabolism, genetic, working on plant fungi symbiosis in cell bacteria. But over a few years, I've gained competence skills in plant mechanism in order to elucidate the mechanism involved in this fascinating interaction. So a few words about this, the mutualism interaction. So as you know, plants attack with a diversity of bacteria and fungi, but only plants present in 4 orders have the capacity to establish endosymbiosis with diazobacteria, leading to nodule formation. We have on one side gram-negative bacteria, rhizobia, have the capacity to establish symbiosis with 2 families, with legumes and Paraspermia genus from the rosal order. And in the other side, we have Frankia, which is an actinobacteria, getting the capacity to establish symbiosis with a large number of families distributed in 3 orders. And this symbiosis named actinoresal symbiosis, and it is what my models. So this bacteria is really interesting because these bacteria have the capacity to fix nitrogen in diazovesicle, and thanks to this fixation, this bacteria can transfer the ammonium and its equivalent to the plant in exchange to carbon fixed by the photosynthesis. So we have a really mutualist interaction, very important for the ecosystem. So a little bit word about these special cells. So diazovesicle cell, so Frankia is able to do a cell differentiation in the tip of IFA and form a diazovesicle with a size of around 2 micrometers. And this vesicle have the capacity have in this world multiple layer of lipids, and this multiple layer of lipids are composed by opanoid lipids, and this opanoid have an important role to not letting oxygen to enter inside the cell, because this oxygen is an inhibitor of nitrogenase. So thanks to this cell differentiation, Frankia, in opposite to most rhizobia, have the capacity to fix nitrogen in culture, in N-free culture, and of course in planta. So in planta, we have here a cross-section of nodule, and you can see here the infection of Frankia in the cortex. So we have cell plant infected by Frankia here. And when we focus on the cell, we can see the diazovesicle, which are larger than we observe in culture. So this suggests that maybe plant also participated in this cell differentiation. So thanks to this nitrogen fixation, actinoresal symbiosis play an important role in the ecosystem by fixing 15 to 25% of nitrogen in Earth. So we have a diversity of actinoresal plants, largely trees and woody shrubs, except for the genus Statisca, which is a perennial herbaceous. They are distributed all around the world, and are really important symbiosis, model symbiosis, in order to understand the evolution of this nitrogen fixing nodule, because we consider that they are the oldest plant symbiosis of this system. They also brought a lot of ecosystem services. Indeed, they support ecosystem services because they are the best of the fusion, of course. They regulated different services like climate, soil quality, nitrogen cycle, carbon cycle. They also provide different services, such as material like wood, and less extent food via agroforestry or medicinal compound. They also they brought cultural service thanks to the magnificent landscape they brought us. In my team, we are working on Alnus model, which is the unique representative of actinoresal symbiosis in the Betulaceae family. This is also the dominant actinoresal tree in the northern hemisphere. And we are working out of the 35 species, we are working on 4 species distributed in different altitudes. We have Alnus glutinosa and Alnus cordata in the lowland, Alnus alcana in the mid-mountain, and Alnus alnobitula in high altitude. Regarding the Frankia species, they appears as distributed appears as a monophyletic group, clustering in 4 clusters, according to the host plant infection, except to the cluster 4, where we found only strain having no capacity to nodulate or they are not efficient strain in the nodule. The strain retrieve affecting Alnus genus were found in the cluster 1E. And in my group, we are interested by 2 genotypes leading to 2 different phenotypes. Indeed, we found in the nodule some strain, SP positive strain, which have we have not the capacity to cultivate this strain in vitro, but in planta, we observe that this strain have the unique capacity to sporulate in planta. So it's really a unique phenomenon that we can observe for this bacteria. And in the other side, we have SP- strain that we can cultivate in vitro. In vitro, they can produce spore, but not in planta. So it's really an interesting question to understand why there are sporulation in one case and not in the other case. So this, we, in our group, in my group, we sequence different genome of SP- and SP positive strain. And we observe that we have a genome reduction of when we in the SP positive strain. So this strain suggests that this strain are maybe obligatory strain as we observe for other endosymbiotic bacteria. So from this model, using this plant and bacteria model, in my group, we have a multi-scroll analysis from the cell to the ecosystem. So in the molecular level, we try to identify gene, protein, and metabolite involved in different processes, such as infection, nodulation, or compatibility. In the organism level, we measure different factors, influencing physiology, morphology, or fitness of both partners. And in the environment, we sampled and characterized different parameters in order to understand the symbiosis in the ecosystem, such as we identify the biogeography of Frankia, or to evaluate the impact of nitrogen fixation in the nitrogen cycle, such as by assessing the microbial diversity and activity involved in nitrification and denitrification processes. So now I will focus only on the molecular level by looking for the process involved in the infection and nodulation. To do that, we use a plant model. So we use Anus glutinosase model and Frankia alnias model. And when these two partners encounter in the environment, there are an exchange of molecules that permit to recognize each other. And after that, we have in the root observation of deformation, leading to calcium spiking induction. And the bacteria will enter in the root cells, leading to the formation of a primordium. And after 21 days, we have a nodule formation, infected, colonized by Frankia. So we did a few years ago transcriptional analysis of the nodule, and we confirmed that the plant is governed by the same common symbiotic signalic pathway that we found in legume. And in order to get an overview of this symbiosis and using the same model, we did an analysis of the early stage of the plant response. So we focus on the very early stage after the early root deformation. So this is the purpose of the article that we published last year in the MPMI journal. To do that, we did a new experiment. You inoculate Frankia in front of Anus glutinosase, and we tested two conditions. So first one, we add the superintendent of culture Frankia cells, and the second one, Peter Pejic, a researcher in my group, developed a new condition by trapping Frankia cell in a dialysis membrane. The purpose of this condition is to permit Frankia to sense the plant and to continuously deliver molecules to the plant. So thanks to by using this post condition, Pascal Fournier, a technician in my group, extract RNA from inoculated roots and non-inoculated roots, and we did microarray transcriptomic analysis using microarray approach. So what we observe, so I focus only on upregulated genes that we found in the two conditions. We observe around 200 upregulated genes, and 20% of these genes are associated to secreted protein. And we can affiliate this different protein to 3 categories. First one, protein implicated in oxidative strain. Some are pathogenic-related protein in the human system. And also we identify protein that responds associated to stress response. So this suggests that after inoculation of Frankia, we observe an activation of plant immune system. So this raises the question about the role of immune system during the symbiosis. That's why we did a novel analysis of all data you get in our group. And Mathilde Vincent, doing her thesis in my group, did bioinformatic analysis by focusing only a gene involved in immune system. And she used the data at early stage, after 2 days after inoculation of Frankia, and also the previous data you get in the nodule. So she confirmed that we have overexpression of this gene associated to the 3 categories, but also a 4th category was identified by Mathilde. Indeed, we observed gene associated to the phytohormone production. So this suggests that the plant induces after infection or in the nodule the systemic acquired resistance. We also observe, as you can see here, that in the nodule, we have less gene overexpressed. And you didn't see here, but we can see also that we have not the same gene overexpressed in the nodule compared to the first step of infection. So this suggests that the plant active a plant defense in the nodule, but in a basal level. But we have to confirm that. We also observe that some gene, and the most overexpressed gene in the early infection, which is NSLTP, is also overexpressed in the nodule. So we really interested by this gene, and we did focus on analysis, functional analysis of this NSLTP. So a few words about NSLTP, which are non-specific lipid transfer protein. They are an antimicrobial secreted peptides getting a signal sequence with a matrix sequence harboring A-cysteine. These A-cysteine are important to form disulfide bridges in order to have an hydrophobic cavity to bind to different lipids. In the literature, these NSLTP are considered to be involved in different processes, such as resistance to biotic and abiotic stresses, to sexual reproduction, or to membrane formation. So Melanie Gasser, which is with us, and did her thesis with me, focused her thesis by analyzing this LTP through 3 axes. She did the first axis by localize this LTP in different tissues, by testing, evaluated this effect of LTP on Frankia culture, and also she carried out a broader analysis of this family of NSLTP nodulating and non-nodulating plant in order to trace his evolution. So first step and first objective for Melanie, she did use a water section after contact with Frankia when we observe the deformation of water, and apply anti-AGLTP24 antibody, and as you can see here, that we observe small spots around the roots, and when we focus on those spots, we can see that AGLTP24 is co-localized with the root hair deformation. So this suggests that the plant address specifically this peptides in the site where the Frankia will enter inside the roots. So maybe this LTP is involved in the Frankia infection. Next, we did the same analysis, immunolocalization analysis with the help of Séverine Balmain. So we did the cross-section of nodule, so in the last stage. And here we have the negative control, and here the positive, you have our nodule with applied with anti-AGFDP24 antibody, and we see that the fluorescence in plant cell infected by Frankia. So when we focus on this plant cell, we can see that the AGFDP24 is colocalized around the vesicle. So here we can suggest that these peptides are addressed specifically to nitrogen fixing the vesicle of Frankia. We would like to go further and to test the effect of these peptides on Frankia. In order to do that, Melanie developed a new approach, a new biological approach in E. coli to produce this LTP. The purpose is to get this peptide with the spatial conformation of this peptide with disulfide bridges. So after producing this LTP, she applied in Frankia and tested different physiological effects. So by measuring the nitrogen fixation by acetylation assay, and also by measuring aerobic respiration, which is another way to measure the cell viability on Frankia. So we use resazurine bioassay. So by applying this peptide with a gradual concentration on Frankia, we can observe here by on acetylation assay that we have above 100 nanomolar reduction of nitrogen fixation activity on Frankia. And when we tested and measuring the cell viability, we observe that we need a higher concentration of this peptide to observe a negative effect on the culture. So this peptide is an antimicrobial peptide at high concentration. But what's about the concentration that the bacteria will encounter in the plants? So this is a tricky question in order to have an idea about the concentration. So we supposed that the plant have no interest for killing or reducing the nitrogen activity inside the plant. So we would like to go further and to evaluate the molecular response of Frankia and suggest that maybe the plant will produce a sub-inhibitory concentration. So that's why we choose to test this and to evaluate the molecular response of Frankia by using 1 nanomolar of AGFDP24. So to do that, Melanie did the true culture, so grown Frankia with or without this LTP at this sub-inhibitory concentration, extract RNA, and did RNA-seq approach. We observed the overexpression of around 100 genes, and the first category of gene we observe is that we have overexpression of gene associated to energy conversion, aerobic respiration, and nitrogen fixation. And this profile will already observed in other conditions. Indeed, we have already did transcriptional analysis of Frankia in a nodule, and we have the same profile. So this suggests that maybe LTP24 mimic the plant condition, and Frankia sense this LTP as in the plant. But in the other side, we observe also some we observe overexpressed gene affiliated to the stress. Indeed, we observe gene encoding protein, encoding chaperone, and encoding protein involved in the protein refolding or DNA reparation. We also observe OXIR, which is a transcriptional regulator, described in other bacteria as important for stress response. So this suggests that Frankia sense, even we brought the LTP at sub-inhibitory concentration, Frankia sense this peptide as a stress and response accordingly. So in conclusion of the two part, first of all, in the early infection, we demonstrate that EG-LTP24 is co-localized in the root, in the deformed root hair, in the root hair nooks, and we have very few examples in the literature of NSLTP associated to plant, symbiotic plant, but we have one case in the Medicacotron catula where the author identified MTN5, which also the gene encoding this peptide is induced during infection and nodulation. And they construct NSLTP saline roots and observe a reduction of colonization of primordial nodule formation. So we can suggest that the author suggested that maybe this LTP is important for the formation of infection threads, and maybe we have the same phenomenon for alnus with Frankia. In the nodule stage, we demonstrate that LTP24 is co-localized in the vesicle, and the in vitro test we made, we demonstrate that we have an impact of this LTP on nitrogen fixation and on the Frankia growth. We have another example of NSLTP in other legume, in Astragalus cynicus, and also they observe an overexpression of this gene during infection and nodulation. They also did a localization of identified the localization of this peptide in the symbiosome membrane, and construct an SLTP saline mutants, and observe an impairment on nodulation and symbiosome membrane development. They also evaluated the nitrogen fixation in nodule of mutant and observe a decrease. So maybe, such as in this model, our NSLTP could be bind to the lipid that we can find with the nitrogen fixation site in Frankia. So we can suggest that maybe they can bind to opanoids that we found in the vesicle of Frankia, and thereby, through this binding, modulate the nitrogen fixation. So we would like to test this, and we have to test the binding of EG-LTP24 to opanoid lipids. And of course, we have to develop the genetic tools to test and to confirm these different hypotheses. So after these two axes, we turn our attention on a broader question about the evolution of this NSLTP. So the question is, this family, if this family have the same evolution history that we observe for the gene associated to common symbiotic signalic pathway. Indeed, several work done a few years ago that suggests that the ancestors of these four families have the predisposition to establish symbiosis, and during the evolution, we have several loss resulting to the current distribution of plant having or not the ability to nodulate. So to test this, we use 15 plant species distributed in these 4 orders, and having the capacity to establish this symbiosis. So the first thing we have to do is to extract NSLTP in these different proteins, and we have to develop a bioinformatic tool. So to do that, Melanie, with the help of Jean Keller, develop a bioinformatic tool to identify NSLTP. They combine 2 approaches based on the identification of conserved motif. They select only secreted peptides, and keep also only peptide NSLTP getting 8 cysteine. And we have the kit up here. As you can see, you have from the 15 proteome you use, we have more than 700 NSLTP, so around 45 NSLTP per proteome. So this suggests that we have an abundance of NSLTP per genomes, and suggests that plants have extensively duplicated this family to permit their conservation or the divergence of gene function. But what about the NSLTP really involved in nodulation and in symbiosis. So in order to respond to the question, we distinguish, so Melanie distinguished the NSLTP coming from plant having the capacity to nodulate in purple, and the other, the non-symbiotic plant in red, in order to see if a profile emerged. And as you can see, there are no single profile standouts, and indeed we have NSLTP evenly distributed regardless of plant symbiotic status. So in order to go further, you did as a hypothesis that maybe we have to identify the NSLTP overexpressed in nodule. So to identify, we have the information for our model, but not for the other model. So we work over transcriptional data in the database of different plants in the nodule. So in order to have to get the information of the overexpression NSLTP in nodules. And in our model, we complete this analysis by RT-qPCR. So you can see here that in the different 5 models we use, we observe in the case you are in agloca no overexpression of NSLTP. It's really interesting because in this model we have no vesicle formed inside the nodules. Indeed, Frankia fixed nitrogen in this model through IFA. So maybe this suggests for us strongly that there are a correlation between the presence of this cell differentiation and the presence of NSLTP, but we have to confirm that with other models. So if you have other transcriptomic data in other models, we are really interested to collaborate together. So here we observe the overexpression of gene in our model, and also Datisca, Medicago, and Parasponia. So this correlate between the NSLTP overexpression and bacterial differentiation in nodule. So we position these different overexpressed NSLTP in the 3, and supposing that will be a clade together, but not really not. They are really in the different clades. So the fact that we observe the presence of this NSLTP, overexpressed NSLTP nodules in different clades suggests that the plant will recruit different NSLTP in order to respond specifically to the symbiosis. So we have maybe a phenomenon of conversion of function in the evolution of NSLTP. So in order to confirm this hypothesis, we have to do an analysis of the other LTP found in different clades and to see if they are really implicated in the phenomenon of nodulation, such as we did with LTP24. Another perspective also of this study is to study other overexpressed genes that were associated to defense. Indeed, we have 2 categories of gene corresponding affiliated to protein having antimicrobial activity. You have the antimicrobial peptides, such as LTP, and also defensin, but you have also pathogenesis-related protein. And here we have the overexpression of their expression on the early infection at 2 days after inoculation in the nodule. And you can see 2 profiles. Indeed, all PR protein are overexpressed after infection, but not in the nodule. And this suggests that we have an inactivation of PR protein after Frankia recognition, but most of antimicrobial peptides are expressed during the infection and also in the nodulation. So such as LTP24, maybe it will be interesting to study the other peptides to see if there are other functions such as we see for LTP24. And we did one study before, one study before in one defancine, HGD5, few years ago. Indeed, Lorena Caro did her postdoctoral position fellowship in our group a few years ago, and she observed, such as we observe for FTP24, a co-immunolocalization of this defancine in the diazo vesicles. So in the plant, we have in the diazo vesicle a co-immunolocalization of HGD5 and HGATP24. So we have really a cocktail of peptides. So it will be really interesting to test in vitro these two peptides as a cocktail to mimic the condition of nodule and to understand what's happened really in the plant. And maybe we have to test also the other, but we will begin with two. It will be good. So in order to go further, I would like also to extend plant analysis using transcriptomic data sets obtained from other symbiotics interaction, and particularly by using SP positive frankenstein, which sporulating peptide will really be interesting to test these strains. And I would like also to understand their function in other biotic and abiotic stresses. I would like to thank Melanie and Mathilde doing their thesis with me, all the members of my group. I would like to also to thank the different partners who collaborate in this project and all the other ongoing projects. And I would like to thank you for your attention.
Thank you so much, Hasna, for a really fascinating talk. At this
I will
point, I just
open
want
the
to
lights.
remind,
Sorry.
yeah, turn on the lights, it got dark.
Yes.
While we're making the switch over, please post your questions in the Q&A box below. And yes, I'm seeing lots of, Hasna, lots of applause coming up, and also some comments in the chat. Great presentation, really fascinating. And I'm so glad that you were able to present that follow-up evolutionary work to see where it's all going. Okay. Well, while we're waiting for people to post questions, Siva, do you have any of your own? I know you had a lot of ideas coming into this.
Yes. It's great talk, great work. And I mean, I know it's working with cysteine-rich peptides is a little hard because most of them belong to this defensin-like families, and so they all have some level of antimicrobial activity, and at sublethal concentration they might do something. And I'm glad that you tested the transcriptome of the bacteria when you treated with these peptide as well. So inside, it's interesting that even inside the nodule, they are binding to these lipids on the vesicular membranes. You can localize them to these vesicle lipids. And have you tested, like, do they have a structure to bind to any proteins as well, or they are restricted only to lipids?
We didn't test it, but in the literature, you can complete Melanie, but I understand that this family have the capacity, they tested in vitro that they have the capacity to bind lipids. Main lipid, and I have no idea if there are tests about the protein. I'm not sure.
I only saw on literature that NSLTP bind lipids, and I don't know for proteins. And here we don't know if this NSLTP bind bronchiolipids because we have just the localization. We saw that this AGLTP24 is addressed to the nitrogen fixing vesicle, but we don't know which lipid it binds. So it's a question.
It's the question. We have to test this. If they bind bacterial lipid or plant lipid. And we have the both hypotheses.
Just a suggestion for bacteria, we have a lipid arrays where you have like 200, 300 lipids in, and
No,
you
not
get
well, yes, we
some
tested.
some arrays that you can just
Yeah,
put
so
on,
strip, yeah, lipid strip.
and then you can
Yes.
put, yeah.
We test. We have master student did a test with Melanie, and not 100, with how many? 20, 50 lipids? 20? I'm not sure. And it's work for one lipid, but it's not really. We have not the connection with the plant lipid, so really we have to extract lipids.
Lipids
and to be sure that what's really happened. I'm not sure that this will happen, will help us to understand what's really happened. So we could have an idea. So we did this experience, but what we plan is to extract lipid, or to have an equivalent of this lipid bite, and to test it in the lab.
Yeah.
Yes, and the big challenge is to extract the lipid of Frankia, because it's opanoid. So it's very difficult, and it needs a lot of work to extract them. So we think about that, but I was at the end of my PhD. So.
Yeah, great question. And that's always, what we're wondering is, what are the actual molecules they're binding? And at least you now have a really good candidate to test. So, okay, I'm going to turn to the Q&As. We've been accumulating some questions there. And this is just a reminder to those of you who want to ask a question to please post it in the Q&A box at the bottom, and we'll take them in turn. So Pascal Fraté has a question. Welcome. Can the microbial activity of LTPs directed against other microorganisms, so that could infect nodules, your symbiont might have co-evolved to be resistant to this LTP?
So we did also some tests of this NSLTP24 against a few other bacteria, and we testing some bacteria that we can find in the rhizosphere. So we test Pseudomonas and Bacillus. I'm not sure if we test other bacteria, Melanie, but
Micrococcus.
And Micrococcus. And what we see, and we did not publish yet, and that we have an impact on the morphology of cells. Cells are more longer, and we suggest that we stop the division of the bacteria, such as we can see for NCA. Maybe, I don't know, but we see this phenomenon on Pseudomonas and on Bacillus. And on Frankia, maybe it could be occur, but Frankia is a filamentous bacteria, so we can see this aspect. So this is really interesting mechanistic question.
Yes, and now that's one of the things that this has been one of the big questions of the last year, is thinking about how these interactions that we think of as binary, right, how those molecules are affecting all the other microbes that are around. And I think it's going to turn out to be a very subtle and complicated story, but the fact that you can see it right there at the nitrogen fixing vesicles is certainly plays an important role in this one. Okay, so Siva, you can take the next one.
We have again there's another one from Pascal. Can cysteines in the signal peptide suggest different secretion when oxygen is low? I think he's asking about the disulfide bridges might be different when oxygen is low.
If we test without disulfide bridges, is the question? I'm not sure to
understand.
Yeah.
Have you tested like reduced and oxidized versions? Because in NCRs we also have that difference, and in the vesicles we still don't know. We always have the
of course, because we are not the same pH status, or maybe there are an impact of the presence or not of the disulfide bridges of the protein. And we have no idea. We suppose that the protein will act with disulfide bridges, but it could be indeed the linear proteins have an impact also, but we didn't test it. We really test only with this disulfide bridges. So yes,
of course,
it could be really interesting to test it.
Yeah.
And of course, with disulfide bridges, you always wonder there's so many combinations that you can have that give you so many different structures. But I'm sure that's a good structural question for the future. So Xian Yang Wei says, thank you for your presentation. Do you consider that NSLTP is involved in the specificity of nitrogen-fixing bacteria infecting certain plant hosts?
If NSLTP interact with nitrogen fixation? No, I'm not sure to understand.
The question is do you think that this protein, or this, I guess it's bigger than a peptide, do you think that this protein is involved in the specificity between this nitrogen-fixing microbe and its host? So the last question was more about microbiome, and this one is saying, do you think that this could be a specificity factor?
Specificity about the compatibility of the SNL-LTP. What I see, not in my case, but maybe in the literature, I see that antimicrobial peptides could be involved in the compatibility. I'm not sure if I respond to the question, but I can respond like that. We have some antimicrobial peptides, NSF1, NSF2, involved in the compatibility in legume. And NSLTP, I'm not sure to see something in the literature. I'm not sure, maybe, but what I saw is that the implication of the thread infection and the symbiosome formation in legume. So really the membrane formation during the infection and during in the nodule functioning. So we suppose that for Frankia, it will be the same thing. In order to permit the penetration of Frankia inside the cell through the infection threads, the membrane will be formed or to maybe to guide the bacteria inside the cells, because if you see in the pollen, NSLTP is also important in the sexual reproduction of plants, and you can see in the pollen. In the etamine, and it's important in the etamine to guide the pollen inside in order to go to the ovule. So maybe you can suppose also that NSLTP is important to guide Frankia inside the cell. We have these different hypotheses.
It's an interesting question. But we don't have enough information about the function of NSLTP in different symbiosis model. Here we have just one for Alnus glutinosa, but there is no other study in other actinorhizal plants, and there is a few in Medicago and Astragalus. So we don't know if this NSLTP have one function, the same function in the different symbioses, or if it's with the evolution, and this NSLTP is here to have evolved to respond to certain specificity of the nitrogen-fixing bacteria or the symbiosis.
Right. And the fact that you see these independent recruitments of the NSLTPs in different lineages means that they may, in fact, be involved in specificity. But I think you're right. You need to look more closely within the actinorhizal
We have to have
plants.
the function, because here we can saw the maybe a convergent evolution, but we need to have more information about the function to conclude that.
So exciting, though, to finally have a tool where you could really probe those questions now. Okay. What's our next question here? Do you want to take that one, Siva?
DIR1 is an NSLTP from Arabidopsis thaliana, and it interacts with high PRP type LTP. Do you think that different NSLTPs will interact between them? Like we have this like between one LTP is interacting with
Yes.
another one together. Do you think they can act? I think you're testing.
Between two NSLTP?
Uh-huh.
I don't know. I know the last we have there are last publication of astragalus, but not between two NSLTPs. Between NSLTP and the chaperone, the NSG, they demonstrate that there are co-localization of these two proteins, the NSLTP and the NSG, and suggest that they work together in order to modify the lipid composition and to bind to the membrane of the symbiosome. So it's I'm not sure to see other example of co-working of two protein.
I saw in literature the two LTP interacting between them, but not in symbiosis. So we don't know if it's the case here. And we, as Hasna said in the presentation, we saw another LTP and on overexpressed in nodule, but we don't know the localization. So we don't know if they are at the same point at the same time, and if they interact, we really don't know.
So many questions this study raises.
Yes.
Lots of next steps. So Andre Laroche says, very eloquent work. How difficult was it to develop an antibody specific to the ulmus LTP?
24?
Yes.
It's only we bite. We did it by not directly by the peptides, the anti-peptides. We only use a small sequence. We synthesize small sequence of the peptide, and after we order antibodies against by using those small sequence, and to test it to be sure that they are specific against our proteins. In vitro.
How long was the segment? Did it have a couple of the cysteines?
To have the antibody, or
in the section that you sent for antibodies? Did it have the cysteine? Must have. Yeah.
Not have the sequence we used. You know, I'm not sure what we did. We write it in the article. I'm not sure what we used.
Oh, that's okay. I was just wondering whether
I don't remember.
No.
Sorry,
I don't remember either.
Yeah, I was just wondering with the. I mean, it's wonderful that it works with a small piece, and I was just wondering how that works with the folding of those cysteines. How that would affect the peptide.
Yes.
Very cool.
There's one more from Nicholas. Do you think that NSLTP could be implicated in membrane contact sites?
In the membrane contact sites between the plant and the bacteria.
And the bacteria, yeah.
We supposed, based on the literature, that during the affection, the LTP will be localized in the symbiose, in the infection trees. So we suppose that is in the plant site, in the plant membrane. In the nodule, we are not sure if it is the vesicle. It's around the vesicle, but it could be also the membrane around the vesicle. We are not sure because it's not sufficient. We have to do confocal microscopy to be more precise, or immunogold, such as the other work was done with the LTP in astragalus, to be really sure the position of the LTP. So in the nodule, we are not be sure if it's really vesicle or the membrane which capture which surround the vesicle. So So it's why we have to test the lipid from the plant and the lipid from the bacteria. Also, it's an indirect way to respond also to the question.
I guess the other question is, when the Frankia is first encountering the root hair, right? You see this,
Yes.
the LTP right on that surface. And is that a point of contact? Interesting.
And you have to know, it's why I am I'm trying to say if I'm really interested for other models, because this deformation of root hairs is specific of fungal order and not in the order, so not in rosal and cucurbita. So we have an intercellular entry of Frankia inside the cells. So maybe there will be another LTP or other protein involved in this infection. So we are really different way of enter of bacteria inside the cells, and maybe it's why it's really interesting to see if there are really a conversion of function in order to have the same purpose.
So we have another question that's on essentially the same topic by Juan Jose Salazar Cortez, who says very nice work, and he is also wondering, excuse me, about the mechanism of binding. Like, is there a specific receptor, or is there a specific lipid on the surface of the bacteria? And I guess that's the big question.
Specific receptor
yes,
in the plants,
or
in the bacteria, on the plants.
On the bacteria.
I think on the bacteria. So if you have the plant LTP, is there a receptor on the bacteria, or just even a specific lipid?
We didn't see the same experience we did. We have also an article published by Peter Pujic in 2019. He identified the overexpression of string in the membrane tubes. So we have the reciprocity of this experience. And what he saw, he didn't observe the overexpression of receptor in the bacteria, but he observed the overexpression of gene involved in the cellulase, encoding cellulase, and this suggests that maybe Frankia encodes cellulase in order to degrade plant tissue in order to permit the entry to enter in the cell. And he also observed cellulose synthase, and he suggests that maybe the bacteria will what's the name? Stiffen the IFA tips in order to penetrate inside the plants.
like any antimicrobial peptide would do, like it can maybe open up the membrane just because of the concentration of these peptides being present on the membrane.
Yes, really. But I didn't remember that that some receptor are
in our second article, when we test sub-inhibitory concentration of LNS-LTP, and we look at the bacterial response, molecular response, we show some ABC receptor activated when we put NS-LTP, but we don't know because there is no molecular tool to study Frankia, so we don't know if these genes are really ABC receptor or other stuff. So it will be interesting to know, but the bacteria
like a
little bit difficult to study.
Many of
in Rhizobia, we when we see the overexpression of this transporter, we say maybe it's like big in Rhizobia, but we have to test it.
And, so many. I mean, the fact you've come so far with an almost completely intractable microorganism. Usually the microbe is the easy side and the plant is the hard side. And of course you have it the other way. well, not that a woody plant is ever easy, but so I think there's lots of interest. I mean, that whole question of what the LTP is binding or interacting with in the microbe or on other parts of the plant is a really interesting open question. And I think there's going to be a lot of interesting biology there that you uncover. So we are seeing lots of nice comments in the chat from people who really enjoyed the seminar. And I just want to remind people that this is recorded, and the recording will go up probably either later today or by tomorrow. And so please encourage people who missed the talk to come back and watch it. And I hope that this will give people who want to see a little more detail to turn to the paper, dive in, look at the methods, see the things. And remember, when you ever look at a paper, there's always the contact information for the senior author or the corresponding author. And that's really an opportunity for you to continue this conversation, to ask questions, to email Hasna Boubakri, and follow the conversation a little further. So I think we'll stop there. Thanks so much, Melanie. It's nice to see your little feline friend there.
Thank you.
Watching with us. It's been a pleasure to host you. And I'm looking forward to seeing what some of the next steps in this are. So thanks, Hasna, and thanks, Melanie, for joining us. And thanks, Siva, for co-hosting with me.
Yeah, thank you so much. It was absolutely
Thank you.
Thank you very much.